Welcome back to Crisp Biology. So in today's video we are going to learn about the functions of the tubules. So as you all know, in the structure we came to know about the proximal convoluted tubule, about the loop of Henle, about the DCT and about the collecting duct. So in the next video about the urine formation, we got to know about how the urine is formed by three processes: glomerular filtration, tubular reabsorption and tubular secretion. So in today's video we are going to see how this tubular secretion and tubular reabsorption actually happens, right? So in the proximal convoluted tubule, so this proximal convoluted tubule or PCT is lined by simple cuboidal brush border epithelium. So wherever there is brush border epithelium, it is just to increase the surface area. But what is the necessity of increasing surface area? This is the major site for absorption, for reabsorption of all the essential nutrients and 70 to 80% of water and electrolytes, right? So this is the major site for reabsorption. You should remember that PCT is the major site. So what all does it reabsorb? It reabsorbs bicarbonate, water, sodium chloride and nutrients like glucose and amino acid. What does it secrete? It secretes hydrogen ions, potassium ions and ammonia. So I already told you in the previous video about tubular secretion. If you haven't watched the previous video, please do watch the previous video on urine formation. Next structure is the loop of Henle. So the loop of Henle consists of two parts. One is the descending limb and another is the ascending limb. So the descending limb is nothing but a thin structure, tubular structure, while the ascending limb consists of two segments, the thick segment and the thinner segment, right? It has a thick segment and the thinner segment. So this loop of Henle has minimum role to play in absorption, right? So it has a minimum role in reabsorption. But what is the main function? Its main function is to maintain the medullary interstitial fluids osmolarity. So we will learn about concentration of urine and we will learn how LOH plays a role in concentration of the interstitial fluid. But before that, you need to remember that descending limb is permeable to water and impermeable to electrolyte. So if the water is coming out, what will happen to the concentration of the filtrate? It will increase. So as it comes down the descending loop, the concentration of the filtrate increases, while the ascending limb is impermeable to water. It does not allow water, but it allows exchange of electrolytes like sodium chloride and urea. Here sodium chloride comes out of the thin segment while urea enters the thick segment, right? But it is permeable, that is important. Electrolyte permeability is there, but water is impermeable. In which limb? In ascending limb, right? So if electrolytes are permeable, what will happen again to the concentration? It will get diluted because water is impermeable and electrolytes are getting exchanged. It is coming out, so it causes dilution. Next structure is the distal convoluted tubule. So this distal convoluted tubule plays an important role in what is known as a conditional or a facilitated or also known as facultative reabsorption. Reabsorption of what particular things? Of mainly sodium ion and water. So what else does it reabsorb? It reabsorbs chloride and bicarbonate as well. Again, there is tubular secretion in DCT also. What all will get secreted? Hydrogen ion, potassium ion and ammonia. Next, coming to the collecting duct. So the collecting duct is a structure which extends from the cortex till the inner part of the medulla. So we know this will end in the renal pyramid, further into the calyces. So this structure is very long and it collects this filtrate from various distal convoluted tubules, that is from various nephrons. So this collecting duct is mainly by conditional reabsorption of water based on ADH, antidiuretic hormone. We are going to further see what is the role of ADH in the mechanism of concentration of urine in the upcoming video. So this main structure or main function of the collecting duct is nothing but reabsorption of large amounts of water. So it also secretes small amounts of urea into the medullary interstitium. For what? For maintenance of osmolarity of the medullary interstitium. So medullary interstitium is nothing but the external environment, whatever except for the nephron, whatever space is around the nephron is called the medullary interstitium. So into that it will excrete, or it will give out some urea, right? And there is little amount of tubular secretion as well. Here there is secretion of H plus and potassium ions. So this was about the functions of the tubules. So in the next video we are going to look about how the concentration of urine happens. Please do share, like